Welcome to Part 2 of Chapter 5, Microbial Metabolism. In this section we're going to discuss carbohydrate catabolism, lipid and protein catabolism, and the biochemical tests that are useful in identifying bacteria. Under the category of carbohydrate catabolism, we're going to cover glycolysis, the Krebs cycle, also known as the citric acid cycle, electron transport chain or ETC, anaerobic respiration, and fermentation. Glycolysis means sugar breaking. Lysis for breaking, glyco coming from sugar or glucose. When researchers were first figuring out how organisms, from you on down to bacteria, make use of complex sugars to get energy, they studied glucose because almost every organism can make use of glucose. Other types of sugars, such as lactose and sucrose, may not be used by the organism, depending upon whether they have the enzymes or not. But all organisms have the enzymes to deal with glucose, or almost all of them anyway. As we go through glycolysis, the things that I want you to remember are what energy molecules are either produced or consumed, what the beginning substrate is, and what the ending product is. Also, there will be a few intermediate products that I want you to remember because they're important in other pathways. Let's start at the top here. The beginning substrate is glucose. That's why we call it glycolysis. And glucose is a 6-carbon sugar. Each of these blue balls represents a carbon. Now the first thing we do is we take an ATP and we put one of the phosphates from the ATP onto the glucose. Remember, this is called substrate level phosphorylation. Now you're probably thinking, why in the world would we use up an ATP when we're trying to generate energy from this process? Well, like any good business, sometimes you have to spend money to make money, or in this case, you have to spend energy to make more energy in the end. So we use an ATP here and another ATP here, putting a phosphate group on both ends of the glucose, and things get rearranged so that you end up with fructose 1,6-biphosphate. And this is the energy requiring steps. Now the next step is the lysis part, where we take the 6-carbon sugar and we split it into 2 3-carbon sugars, both of which have a phosphate group on them. One of them is dihydroxyacetone phosphate, or DHAP, and the other one is glyceraldehyde 3-phosphate, or G3P. Now in this figure they left out the 3, but I have a tendency to put the 3 in. Now, for the rest of glycolysis, you just need G3P. So the DHAP, the one DHAP is converted to G3P so that from here on out you've got 2 G3Ps. Okay, remember that. So here's where we start making energy. Okay, we take an inorganic phosphate group. It is put onto the G3P, and a hydrogen is transferred from the G3P, NAD+, and we generate 2 NADHs from that. These are going to generate energy later. Okay. Then from there we take one of the phosphates and we put it onto an ADP, and we make ATP. Now because there's 2 of these, we make 2 ATPs. We've just broken even. We've used up 2 ATPs, now we've made 2 ATPs. We do some other funky stuff, and we take the last phosphate group, put it onto an ADP, make another ATP. Because there's 2 of these, we make 2 ATPs. So we've generated 2 ATPs net. Now the final products are 2 pyruvic acids. Sometimes I'll call them pyruvate. It's the same thing. So you start out with 1 glucose, you end up with 2 pyruvic acids, and through the process of substrate phosphorylation, we consume 2 ATPs and generate 4, and we generate 2 NADHs. Now the pyruvic acids generated by glycolysis still have a lot of energy in them. So we don't want to throw that energy away. This cell is very efficient. So we're going to continue on and we're going to get some more energy out of it. So we're going to slice off an acid group, and this is going to come off as CO2. Remember that a carboxyl group or an acid group is a carbon with 2 oxygens and a hydrogen. Now we don't want to throw away that hydrogen. That hydrogen has energy. So we're going to take that hydrogen and we're going to put it on an NAD plus and make NADH out of it. This is going to go make some energy later. So we're going to end up with an acetyl-CoA. The CoA comes from coenzyme A, it comes on there and it transports it into the Krebs cycle. Now, when we're counting up the energy molecules that are produced, we always go per glucose. So because there's 2 pyruvic acids produced per glucose, we count this as 2 NADHs that are formed by acetyl-CoA formation. The acetyl-CoAs then go into the Krebs cycle or the citric acid cycle. So the acetyl-CoAs are attached to oxaloacetic acid, which is a 4-carbon acid. You add the 2 and you get a 6-carbon acid. Now from there we do some rearrangements and we chop off an acid group. Remember, from acetyl-CoA formation, when you chop off an acid group you get CO2 coming off. Now we want to save that hydrogen that's on the carboxyl group. So an NAD plus comes in, picks up the hydrogen. We do that again in the next step. We release CO2, form an NADH. Then we form ATP in this step through substrate level phosphorylation. Then FAD comes in and picks up 2 hydrogens. Then up here we need to pick up another hydrogen, so NAD plus comes in and picks up a hydrogen and becomes NADH, forming oxaloacetic acid. This is why it's a cycle. Our starting product, one of our starting substrates, is also the ending product. And we also add in acetyl-CoA. So now remember, we get 2 acetyl-CoAs per glucose. So we get 1, 2, 3 times 2, so 6 NADHs, 2 ATPs, and 2 FADHs per glucose for the Krebs cycle. Another thing to consider from acetyl-CoA formation and the Krebs cycle is this is where the CO2 that you exhale comes from. This is also the CO2 that's released by almost all cells. This is where it comes from. So I bet you were just dying to know why you needed to exhale. Now on to the electron transport chain. The glucose in and of itself has all been broken down into CO2, no more energy left in it. We've made as many ATPs out of it as we can through substrate level phosphorylation. Now we need to take the NADHs and FADH2s and convert them into ATPs. It's kind of like exchanging money. If you go to a foreign country and you come back to the United States, you take that money and you exchange it back into US money where you can use it. So an electron transport chain are a series of enzymes that are embedded in a membrane. Okay, and these enzymes move electrons down the chain. Okay, and remember, these electrons have energy. Each time you transfer it, you release a little bit of energy, but you also do work. So here, NADH comes into the first enzyme in the chain and it donates a hydrogen. Now this hydrogen is split by the enzyme. The electron is kept by the enzyme complex and the hydrogens are pumped to the other side of the membrane. Now hydrogen, being an ion, can't come back. Those protons cannot come back. They want to come back because, as you'll notice as we go along, we're going to develop a whole lot of hydrogen ions out here, and because of diffusion they want to come back to an area of lower hydrogen concentration. Okay, so the NADH comes in, donates the hydrogen, it gets split, the electron gets taken by this enzyme complex, and the hydrogen, the proton, gets pumped to the other side. Now this electron gets passed to another enzyme complex which uses that energy to pump hydrogen ions to the other side. We're building up this concentration outside here. So then we come to the next complex. We pass an electron over. Now FADH donates some hydrogens here. Okay, so Those hydrogens are pumped to the other side, electrons are donated, keeps going down the chain until the poor little electron is all worn out. It's been used up as far as energy, so now we got to get rid of it. So what we do is, in aerobic organisms like yourself that breathe oxygen, oxygen comes along and picks up that electron. Now, when oxygen picks up the electron, it gets a negative charge and it attracts hydrogen ions, protons that are floating around, and we form water. This is why you breathe in. This is why an awful lot of organisms need oxygen. Okay. Now let's go back to these protons that are on the other side of the membrane. Remember, they want to get back. There's a whole lot of hydrogens on this side, not a whole lot of hydrogens on this side. They'd much rather be back in here, but they can't get past the phospholipid membrane. These phosphate heads, these charged protons kind of get hung up on there. And besides, they can't get past the hydrophobic interior. That's why you got to have a membrane to do this. We call this a proton force or osmotic pressure. It's also called chemiosmosis. Well, what happens is we have an enzyme here called ATP synthase that opens a hole in the membrane for the hydrogens to come through. Now this is just like water coming over a dam. We open up a hole in the dam so that the water can come through and turn a turbine. This part of the enzyme actually turns. We've looked at electron micrographs and they actually turn, and they take ADPs and inorganic phosphates and attach it and make ATPs. Okay, so the electrons are used as energy to produce this osmotic pressure, this chemiosmotic pressure on the other side of the membrane. All of these protons are over here. They want to get back in. We let them in through AP synthase, and we produce ATPs out of it. Now this is not substrate level phosphorylation. This is called oxidative phosphorylation because of the oxygen. Okay. Now you can make 3 ATPs approximately from each NADH. For each of those hydrogens that are pumped out, when they come back through, you can make 3 ATPs. Now FADH2, because it's put into the chain lower down, you produce only 2 ATPs. So when we set things up, when we're looking at how many ATPs that you can make through oxidative phosphorylation from the NADHs and the FADH2s that are produced through glycolysis or acetyl-CoA formation or the Krebs cycle, we do that kind of math. For each NADH, you get 3 ATPs. For each FADH2, you get 2. Now, for most of these enzyme complexes, I don't expect you to remember them, but I do want you to remember cytochrome C. You'll see that it has shown up in 2 different places in the electron transport chain. It's important for a number of different reasons. One of them is we use this to determine a relatedness between organisms. We'll look at the genes that code for cytochrome C, and we'll look at how much they've changed. The reason for this being, you can't get very many changes in cytochrome C without losing the ability of the enzyme to pass electrons around. So it changes very slowly. So it's a good indicator of relatedness. Another interesting side note, in humans, and well, in most organisms with mitochondria, cyanide will bind to cytochrome C and will not it won't be released by the enzyme. And this causes cytochrome C to hold on to the electrons that it gets. Doesn't pass it on to the next one in the chain, and it can't accept an electron from further down the chain. Gums up the whole process. Shuts down the electron transport chain. Well, why does that kill you? Well, it's because you're not making all of these ATPs. All told, from glycolysis down through the electron transport chain, in prokaryotes, you make 38 ATPs. In eukaryotes, you make approximately 36. We lose a couple of ATPs moving acetyl-CoA into the mitochondria. Actually, you're moving pyruvate into the mitochondria. But if you only count the the ATP that we get from substrate level phosphorylation, we only get 4. So there's 30 plus electrons that you're not getting if your mitochondria are not able to make ATP through oxidative phosphorylation. So humans can't live on just those 4 ATPs. We can't cut back our metabolism, and so we die. Other organisms can cut back. If they can't use their electron transport chain, they can cut back their metabolism, they can slow down their growth, and live on just those 4 ATPs. We'll be talking about those organisms when we talk about fermentation. Not all organisms use oxygen for their electron transport chains. Most of the organisms that originally we were familiar with do use oxygen, and they're the easiest to grow in the lab because you don't have to keep them in an oxygen-free environment. But we have found that there are organisms that are anaerobic. They either don't need oxygen or oxygen kills them. They still undergo respiration. They still have an electron transport chain. So what do they use for the terminal electron acceptor? You still have to get rid of this electron, because if you don't get rid of this electron, then you can't get another electron put in, and it can't get passed from enzyme complex to enzyme complex, and the whole thing gets gummed up, stops. So you have to get rid of this last terminal electron. So what do they use instead? Well, my favorite example are the organisms that use sulfur. Sulfur comes and picks up this electron, and because it has a negative charge, just like the oxygen, it picks up those extra protons, and you form hydrogen sulfide, or rotten egg gas. That's one of the reasons why when you're in a swamp and you see those bubbles coming up from the mud and it really stinks like rotten eggs, well, that's coming from anaerobic organisms living in the mud. Now let's talk about fermentation. I alluded to that when I was talking about how cyanide kills you by shutting down the electron transport chain. Well, let's talk about how some organisms deal with either not having an electron transport chain, or what they do when they don't have access to their electron transport chain. So let's take brewer's yeast for an example. Normally, yeast prefers to grow in an oxygen environment. Glucose goes through glycolysis, it goes through acetyl-CoA formation, citric acid the citric acid cycle, and through the electron transport chain. They can grow faster, divide more frequently if they're getting their 36 ATPs. But if you cut them off from oxygen, brewer's yeast will still survive. Here's what happens. It continues with glycolysis. Okay, we're still producing the 2 ATP net per glucose. But since we're not going to send the NADH through to the electron transport chain, because if you don't have oxygen, you don't have the terminal electron acceptor, and so you can't get rid of these NADHs and make ATP. But you need NAD plus for glycolysis, so you can still continue to make your 2 ATPs net. So what yeast does is it takes pyruvic acid, clips off an acid group, forms CO2, and you form acetylaldehyde. Then you take your NADH, you put a hydrogen onto there, and you make ethanol. This is what happens when whether baking bread or making alcohol, you produce CO2 and you produce ethanol so that you can get these NAD pluses that can go back up here to glycolysis and you can make your ATP. Now, in alcohol fermentation, if you're making wine or beer, you're really not after the CO2. That's a side effect. That's why wine and beer are bubbly. But we're really after the alcohol. When we're making bread, we're actually after the CO2. Fluffs up the bread, you get alcohol as a side product, and that burns off during the baking. Now, there are other organisms that do things a little bit differently. Take your organisms, your lactic acid producing organisms that we use in yogurt and cheese making. Start with pyruvic acid, because these organisms have gone through glycolysis. They're forming their 2 ATP. They're forming NADH, or in this case, most of them don't have an electron transport chain. So they still need those 2 ATPs. So what happens is they don't cut off this acid group. They don't produce CO2. That's why you generally won't have bubbly yogurt. So we take the NADH, donate the hydrogens to the pyruvic acid, and we make lactic acid. The lactic acid curdles the milk, and we either get thick milk, yogurt, sour cream, or we pull out some of that and remove the water and we get cheese from it. And that's where fermentation comes from. Now, lactic acid fermentation. Now, we're making use of the fermentation process for our own culinary needs, but the organism's goal is really to make the NADH for this part of glycolysis, so it continues to make those two ATPs. Once researchers figured out how organisms got energy from glucose, they moved on to other macromolecules. Not all organisms live just by glucose, your brain cells being a notable exception. But most organisms use complex carbohydrates, they use lipids, they use proteins as energy sources to make ATP. This chart shows the process by which organisms get energy from complex carbohydrates. So complex carbohydrates are broken down into sugars. Most of them go into glycolysis. Okay. From glycolysis, they go through acetyl-CoA formation, through the Krebs cycle, and on through the electron transport chain, or into fermentation. Proteins, on the other hand, are broken down into amino acids, and they can enter this process at various points. Some of them get converted into pyruvic acid and then they go on through. Some of them get converted to acetyl-CoA, and some of them enter into the Krebs cycle. Lipids undergo a similar kind of thing. Lipids have their glycerol snipped off, and glycerol gets turned into DHAP, remember that? And it gets converted into a glyceraldehyde 3-phosphate, or G3P. Fatty acids, on the other hand, are clipped off into 2 carbon groups. Remember, fatty acids are these huge hydrophobic chains, and they get clipped off and they end up as acetyl-CoA, and they go through the Krebs cycle and on through the electron transport chain. That's how cells use lipids and proteins and complex carbohydrates. Now, back before we had DNA technology, to identify bacteria, we would use biochemical tests. In fact, we still do. We prefer DNA methods, but we still use biochemical tests to determine the ID of the bacteria, because the biochemical test tells us what enzymes the organism has. Once we know what enzymes it has, then we know what genes it has. Now you're probably going, well, how does a biochemical test show us what enzymes there are? Once we know the process of going from glycolysis down to acetyl-CoA formation, through the Krebs cycle, and through the electron transport chain, or if it doesn't do any of those things, if an organism strictly ferments, then we know what enzymes it has. Okay, here's an example. Here's a series of test tubes that have just one sugar in it. Okay, and we have these little tubes in here upside down to catch any gas. So this, and we've also put an indicator in so that we can see if there is a drop in the pH, if there's acid being formed. So this tube here, the pH didn't change. There was no gas formation. There's no cloudiness. The organism didn't grow. So this organism doesn't have the enzymes to process this sugar. We know it doesn't have those enzymes. Here we see that the organism grew and it dropped the pH a little bit, so that we know that it doesn't prefer to ferment that particular enzyme. It prefers probably to go all the way through acetyl-CoA formation, Krebs cycle, onto the electron transport chain, except for it's not producing any gas. No gas up here. Here, this organism produces gas, and it also produces acid. This one produces acid, produces a lot of gas. We can go back to the biochemical processes, these metabolic processes, and determine from the products of these biochemical tests what enzymes the organism has, and from there we can figure out the genes. Okay, that was a whole lot, wasn't it? Okay, once again, don't be discouraged if you didn't get it the first time. This is an awful lot of chemistry. Make sure you go through this multiple times. Let me know if you want me to go over this in lecture in more detail.